Hello! Over the 20 minutes that follow, I will give an overview of how small molecules, and in particular those from natural sources, could be used against arthropod-borne infectious disease. And to highlight and support this statement, I would like to share with you our preliminary results on challenging chikungunya virus with acetylphloroglucinols that we isolated from hops. Before to go on detail of those results, I would like to make some reminders about the general terms of arboviruses. I will mention some notions about natural products, and then I will finish my talk with the results that we had on chikungunya. Arboviruses, by definition, are disease-causing viruses transmitted through the bite of hematophagous arthropods such as mosquitoes and ticks. Around 4,000 viruses are described up to date. 5 to 600 are transmitted by arthropods, half of them are transmitted by mosquitoes, and around 100 are pathogens for humans. As you may see here in the map, arthropods are predominantly present in subtropical and tropical areas. Chikungunya was identified for the first time in 1952 in Tanzania. In 2005, there were two important outbreaks. The first in Kenya, and the second was the outbreaks that happened in the Southwest Indian Ocean islands, in particular the one of the La Réunion. 2 years later, chikungunya arrived in Europe. There was reported cases in Romania, Albania, Italy, and 2010 south of France. In 2013, chikungunya arrived in the region of Caribbean islands and later was spread around Americas. Alphavirus is part of Togaviridae family. In this family, we have two genus, Alphavirus and Rubivirus with Rubella virus or the German measles. The clinical manifestation of this virus is completely different. I won't go more in details. Just to mention that the German measles is part as well of a Togaviridae family. Going back to alphaviruses, we have two groups depending the geographical distribution, the New World one and the Old World group. The New World will have equine encephalitis, Eastern, Western, and Venezuelan, with the animal reservoir will be birds and horses, and the vector would be Aedes aegypti and Albopictus, with the clinical manifestation will be encephalitis. In the second group, where chikungunya is taking part, we will have Ross River, Mayaro virus, Mliki forest virus, and Sinbis virus. We have as animal reservoir more primates and mammals, same vectors, and this time the clinical output will be more fever, rash, and arthralgia. From a genetic point of view, chikungunya virus is around 12 kilopair based viral genome, single-stranded positive sense RNA, which make him part of the group 4 of Baltimore system, 70 nanometer in diameter, and we have two open reading frames. One that will give the non-structural polyprotein with the replication complex, and the second one that will give the structural proteins with the nucleocapsid and E1, E2 glycoproteins that will give the envelope of the virus. We know today that the matrix remodeling associated protein 8 is the receptor responsible for the entrance of the virus on host cell. It's a clathrin mediated endocytosis, pH dependent. Once the viral genome is liberated in the host cell, we will have the translation of this replication complex in first step, and in second step we will have the synthesis of a minus stranded RNA, and there will be two main functions. The first will be to synthesize, to give more positive stranded RNA for the formed virions, and the second function will be to the transcription of a subgenomic messenger RNA that later on, after translation, will give the nucleocapsid and the two glycoproteins. In 2005, as I mentioned, there was an important outbreak in the La Réunion island, and what happened is that in the E1 glycoprotein, in the position 226, the amino acid alanine was replaced by a valine, and this had a huge consequence for chikungunya distribution because Aedes albopictus became as well a suitable vector for the distribution of chikungunya. This brought as well two major consequences: that the virus increased his ability to infect host cells, and as well to survive for a longer period in the environment. I would like to remind you as well that alphaviruses, flavoviruses, rhabdoviruses as well have highly efficient self-amplification of RNA in host cells. What it means that, well, it's used in gene therapy as advantage to use them as a vector. And comparing with the conventional messenger RNA manufacturing, fabrication of vaccine, what we will have here, that with we will have the NSP complex next to the gene of interest. The vector will enter the host cell like with the conventional messenger RNA, and will recognize the toll-like receptor on the endosome. Then we will be the activation of the two helicase RIG1 and MDA5, and at the end of the day, what we'll have an increased number of MHC1 complexes that will be expressed in the surface of the plasma membrane of these infected cells. And at the end of the day, this will increase as well, then the antigen presenting capacity of these complexes will be higher. We'll have higher degree of this presentation, and we will have furthermore an answer of the host defense system, in particular the CD8, an increasing induction of cytotoxic T cells from the T cell adaptive immunity. And as well, we will have more proteins that will be recognized by the antigens in the surface of these infected cells. Another important feature for chikungunya is that this virus is able to activate neutralizing antibodies, which means that, you know, besides this capacity to use the NK cells in the antibody-dependent cell-mediated cytotoxicity pathway, we will have a direct binding, blocking of the virus by these neutralizing antibodies. And what happened, one important event happened last year, November 9th, is that FDA approved the first live attenuated vaccine against chikungunya developed by Valneva, French-Austrian company. But however, besides that, we need still antivirals, because today we are treating yet the symptoms and with anti-inflammatory drugs or analgesics, and we don't have still drugs that are targeting really the virus. Why natural products could be and are a source of inspiration for various kinds of drugs, anti-cancer, antivirus. I wanted to share with you some classical, let's say that way, secondary metabolites used from centuries as drugs. Digitaline, the pilocarpine, histamine derivative, quinine, the anti-malaria drug, and morphine. What I wanted to point here is that we have a huge diversity, huge variety of functional groups on these secondary metabolites used as drugs coming from natural sources. We have as well stereochemistry, really important for the fitting of those drugs, either with enzymes, with receptors, or just to express some physiological response. And we have as well nitrogen, oxygen, really important atoms that will, at the end of the day, provoke and express this need to fit with biomolecules and biological targets. I wanted to remind as well that when we speak about fitting, about activation, about binding, it's always matter of forces of interaction, of energy and enthalpy. And what have natural products, because besides this stereochemistry and configuration needed for fitting well, they have this ability to create electrostatic interactions with biomolecules, dispersion forces, London forces, van der Waals forces, depot-depot induced moments, ion-depot, hydrogen bonding, really important for biomolecules, hydrophobic interactions and PP interactions, and as well creation of covalent bonding and ion bondings. Hops is used in brewery industries from centuries. Here you can see the bractea, the female inflorescence of the hops. Humulus lupulus is the Latin name of this plant. There is two main reasons why we use this plant during the fabrication of a beer. Is because we will bring the flavor, this bitterness of the beer, and the second reason is because the compounds that I will present you in some slides just in a minute, they have these antibacterial properties, and by being aqueous beverage, well, you can imagine that the spoilage by presence of no desired microorganisms, bacteria, will bring some, will decrease the quality of the beer. There is several group of compounds, essential oils, flavonoids, and so on, but I would like to bring your attention more in those acetylphloroglucinols, or alpha and beta acids as they are known. We have 6 members in the 2 groups. They are quite similar in their from chemical point of view. The main difference between the alpha and the beta acids would be the carbon, the hydroxyl group in the position of this carbon from the 6 ring element in alpha acids, and this, in the same position we will have, in the case of beta acids, isoprenyl radical. This is the first feature. The second feature will be the length of this short aliphatic chain, and his ramification and the presence or the absence of one more methyl group. As I mentioned, the antibacterial properties of those molecules are known and are the reason why we are using hops in the brewing industry. In the 40s, there was a paper published, research made by the Institute of Brewing Research Scheme, that explained how these compounds, for the first time, explained how these compounds have this affinity to bind with the manganese bivalent ions, compounds, and as well this capacity of those compounds to make like a hose surface, a ionophores in the cell membrane of the bacteria, and as a consequence to destroy those these bacteria. What we made is a PRNT, a plaque reduction neutralization test, in vitro test. Basically the idea is that you will challenge the virus cells, the model that we used, in different configuration, either the virus alone, either the virus in mixture with the compounds to test, or you will challenge firstly with the compounds, and then you will add the virus, and this will give you different scenarios that will bring some information of what step of the replication cycle of the virus could be inhibited. We have 4 different possible scenarios: virucidal activity pre-treatment, inside co-treatment, or post-treatment. And what we had that in the post-treatment assay, we managed to obtain 80% inhibition by beta acids, alpha acids, cohumulone, which is the major alpha acid, and xanthomimol and flavonoid fraction. Post-treatment assay will indicate more how the inhibition could occur in the end of the replication cycle of the chikungunya, which means the translation, the assembly of the virion inside the cell, and then the leaving from the infected cell. In the literature, there is some information that already reported the potential of natural products to be used as antiviral in the case of chikungunya. Here in the table, you can see mainly flavonoids with their capacity to reduce the NSP1, the NSP3, or the structural E2 protein. We have as well coumarin and terpene. I will just come back to this in a minute. Yeah, it's mainly the post-treatment inside phase as well. What was published in 2017 from the Professor Nates and Lien de Lang in Rego Institute for Medical Research in Leuven, Belgium, is that protein kinase C could be a potential host target for the inhibition of chikungunya virus replication. Just to remind, then protein kinase C is a kinase, is a transferase, could be activated by antigen or signal molecule. We will activate the G-couple protein receptor that will activate the phospholipase C, and phospholipase C will split onto the phospholipids that are in the membrane, plasma membrane, in diacylglycerol and the inositol 3-phosphate. And this diacylglycerol will activate the protein kinase C, and by phosphorylation processes will have a cellular response. In particular, protein kinase C will activate the MAP kinase pathways, 3 well-studied signaling pathways with the extracellular signal regulatory kinase, ERK, P38, and CGen NHT terminal kinase. Here, well, the consequence will be inflammation and apoptosis. There is a key factor here, the transcription factor, nuclear factor kappa B. And this nuclear factor kappa B is responsible for a lot of processes in the cell survival, proliferation, inflammatory response, angiogenesis, and so on. What I wanted to highlight here is that the nuclear factor kappa B is responsible for the differentiation of the T cell from the adaptive immunity, depending on the type of the antigen. We will have a secretion of cytokine and chemokine, and by the presence of different transcription factor further, like ROR gamma T here, we will have the subset TH, more TH Th17, Th1, Th2, and so on. The other thing is that the nuclear factor kappa B will activate as well the maturation, will help the maturation of the dendritic cells, the sentinel cells with macrophage and neutrophil. And we made three hypotheses of mechanism of action. The first was this similarity that was proposed as well by the Forbo, the prostratrine, a compound isolated from L-Forbaceae family, a sponge family. I invite you to read the paper that was published from Mark Littledon in 2019. Yeah, the prostratrine has this capacity to activate protein kinase by similarity with the acetylglycerol, and they identified three pharmacophore functional group responsible for this action, carbonyl and the hydroxyl group, and we made the assumption that cohumulone and acetylphloroglucinols, by having these functional groups in some way similar configuration, could be one of the reasons why we had this post-treatment strong inhibitory capacity. The second hypothesis that we made was based on the antimicrobial properties of these compounds and the lipophilic side, their capacity to integrate the plasma membrane and to inhibit the further development of bacteria or replication of viruses. And the third hypothesis was based on the capacity of these compounds to make a dimer, as was reported similar compounds, fluoroglucinols, described in Leptospermum species. It's the Manuka honey plant, plant from New Zealand and Australia. Similarly to these compounds, we suggest that those acetylphloroglucinols in hops are capable, by similar to Michael addition, form dimers, like form bulky structures like dendrimers, and will inhibit the pathogens as viruses. I invite you to read our papers about chikungunya, and as well we made a further study on Oroposia bunyaviral virus with those compounds. This time we added an in silico studies, molecular dynamic simulation. This was made by Gabriela Lima Menezes. I really want to give credits to these great researchers, Brazilian researchers, that helped me to do this work, and Maria Helena Saivis from São José de Rio Preto Medical University in Sao Paulo, and I'll be happy to discuss with you this study or other questions if you have. This is my email, and I thank you for your attention.